find that that's the right cutoff in order to do gene finding. And I'll just say that this is something that's very unique to PacBio. Other long-read technologies don't have the ability to produce very accurate information from a single read. And short-read technologies don't have the ability to generate reads that are that long. Okay, so I'm going to first give you more information on 16S. So here you can see this is an example of two different runs. So this is a mock community from the sequel 1.0 chemistry from over the summer. And you can see here we have 2.4 million reads. And now with the V2 chemistry that is available in October, we can get 2.6 million reads at the very same time. So if you look here, you can see that the number of average reads per barcode looks very similar. What has changed is that we have updated the 16S protocol, and we will soon release the new protocol. And for this protocol, we've switched from a two-step PCR to a one-step PCR. And this has the effect of reducing the number of chimeric reads that you get during that sample preparation. The second thing is that we changed the barcoding strategy for 16S. And as a result of the new barcodes that we're using for this application, we now have much more even coverage across all 96 samples when you do multiplexing. So now you get an average of 38,000 reads per barcode, but none of those reads will have fewer than 24K reads. And so that's really nice because as a service provider, oftentimes you have to guarantee a minimum number of reads for every sample that you run. And so now this is a lot more predictable. With 16S on PacBio, you see on the left is the expected distribution of species from the mock community, and on the right is what you see from PacBio sequencing. And you see this is pretty even representation, even though 16S does include PCR. And here you see that the results that you get from PacBio sequencing are very consistent across many, many samples. So these are all sequencing with different barcodes of the same sample. So you would expect in an ideal world, these would all be the same. Okay, so what are some of the things that researchers are doing with our 16S technology? This is a publication from last year that was done with Ben Callahan, who is the author of 16S analysis software called Dada2. And Dada2 is very popular for analysis of short-read sequencing data. And Ben wanted to extend this to also look at PacBio data. And what he found was very surprising to him and made us very happy as well. And what he noticed is that, of course, every type of bacteria has a different number of 16S repeats in the genome. And the number of repeats is specific to the species, but the differences across them, whether they're all identical or how much they differ one from the other, is strain specific. And what he found is that by looking at PacBio data, he was able to distinguish all of the different 16S copies. And he could tell which ones were the same and which ones were identical. And that's useful because with that level of resolution, you can actually take that information and then use a reference database, in this case just NCBI, and you can often identify the exact strain that is being sequenced. So in this case, for this mock community, Ben was able to identify that the E. coli used in this mixture was O157H7, which is a strain that was linked to a serious outbreak, actually. And this was really exciting to us because in the past there had been some publications saying that PacBio had systematic errors for 16S. And when Ben took his new software tool and reanalyzed that data, what he found is that the authors had assumed there was only one copy of that 16S gene in the Staph aureus strain they were looking at. But looking at the PacBio data, he was able to see that actually there were 5 different copies. And this became very clear that what the author of the paper had seen was not actually a systematic error, it was the ground truth of different sequences. So moving on to a real-world sample, this is a sequencing study done by the Joint Genome Institute, which is a government research lab in the United States that specializes in understanding microbial life in the environment. And so they decided to sequence samples from a lake, Lake Sakina, that's in Canada. And the interesting thing about this lake is that in this lake, the water in the lake does not mix evenly through the year. In this lake, the layers of water always stay separate. They don't mix. And so they did a study where they sampled the water at different depths and then did 16S sequencing to see how different the microbial communities were in each one of those layers in the water. And so the interesting thing that they found here was that if they used only short read sequencing to understand this members of the community, they were only able to identify the members at the species level about half the time. But with PacBio, they were able to identify the species 74% of the time, so much more consistently. And that's the power of having very accurate long reads. And given the complexity of the lake sample and also most environmental samples, having that level of genus and species resolution was very important. So here you see, this is a chart that's showing the abundance of different clades of bacteria at different depths in the lake. And so each one of these is paired. This is a short read analysis, and then this is the long read analysis. And every time you see one of these gray boxes, Okay. Every time you see one of these gray boxes, it's showing how you are not able to get genus level information just from the short read data, only with the long read data. And as you can see, there is significant information that's lost then if you don't have a high resolution view of your sample. And if I could point out here, for example, in this case, you might think that there were no Elucimicrobia at all in this sample if you were relying on short read data. But if you look in the long read data, you can see that it's actually fairly prominent. So I have a second example to share with you of a human microbiome sample. This was research done in China, actually, by a researcher who was interested in understanding the contribution of the oral microbiome to dental disease. And so she sequenced samples from children that had a lot of tooth decay and also children who had very healthy mouths. And what she found is that when using PacBio, she was able to identify the species for almost every read, whereas using the short read only about half the time. And that turned out to be pretty important because if you look at the genus level characterization alone, the tooth decay and the healthy samples actually look fairly similar. You have the same clades that are the most abundant in each of these. And so you might not think that the oral microbiome had that much of an impact on this tooth decay, but that would be a mistake because in fact, there are very different species that are abundant in children that have high tooth decay versus have very little tooth decay. And so here you have the distribution or what fraction of children with tooth decay had this particular species present in their microbiome. And then conversely, these species in green are showing what proportion of children with healthy mouths have these particular species present in their mouths. And what was most interesting to me is that if you look here at some of these most prominent members, you can see that there's Streptococcus both in the diseased mouth and in the healthy mouth. And it's only by looking at the species level information that you can tell which Streptococcus are the bad actors and which ones are correlated with healthy teeth. Okay, so moving on to our next application, HiFi metagenomics. So this is the performance on the Sequel II system. We did shotgun sequencing of a mock community, and here you can see in green is the expected abundance of these different species in that sample, and in blue you have the abundance that is reported with PacBio sequencing. And you can see that the green and the blue match very closely, even down to very low abundance. So in these species, you have, these are present only at 0.01% abundance in this mock community. And you can also see that the proportion of these reads does not correlate with the percent GC. So this is a very nice feature of PacBio sequencing in general, the very low amount of GC bias. So how much data can you get from one Sequel run? Quite a lot. So this is 9 human fecal samples that were sequenced with the new chemistry, Sequel II E2.0. And you can see that you get a large number of reads here, and that the average read length is up to 10 QB, and the accuracy is very high. So Q40 means 99.99% accurate. So just like Illumina data. And the interesting thing to my mind from this data is that what you see is that the average read length and the accuracy of just the HiFi reads without assembly outperforms the quality metrics that you would expect to see from most short read assemblies. And so even before you attempt to assemble, you already have more contiguous information than what you would have with short reads, and far more accurate than what you could get with ONT. And the other advantage of having such long reads is that you can do gene discovery directly on these. So in this case, these samples had an average insert size of about 8 baby, and you can see each one of those reads has between 6 and 8 genes on it, which can easily be detected using the same pipelines that you would use to do gene prediction using Illumina assemblies. So how about environmental samples? So environmental samples, of course, are extremely complex, and I think that HiFi reads are therefore particularly useful for these samples because they assemble so poorly, right? So the more complex the sample, the harder it is to assemble. And what that means typically is that you're not able to recover genes from those low abundant species. But with PacBio, that's not the case. So we did a collaboration with a company called Second Genome. And Second Genome is interested in mining soil for compounds that can be used as potential therapeutics. So maybe antibiotics, maybe cancer drugs, anything along these lines. And they wanted to understand, can PacBio sequencing help them discover genes more efficiently? So what they did is they made 8 KB libraries, and they did HiFi sequencing on those. And then they did gene prediction, and then simultaneously they assembled those reads into contigs. They then dereplicated the genes so that they have a non-redundant, unique set, and then they placed those genes onto the contigs to help do better gene prediction. So when they did assembly, they got some very nice results. And remember, this is soil samples, so very complex. But nonetheless, in many cases, they were able to get contigs that were very large, and often these resulted in complete genomes. So not closed, but complete. And in many instances, they were also able to recover closed plasmids. So they took this data then, and they did two different tools to do gene discovery. One was FragGeneScan, and the other was Prodigal. And as you can see, both of these methods gave very good results. And from that, they concluded that HiFi reads are accurate enough to do gene prediction using short read tools without any further modification or changes to those settings. So the really interesting thing, though, was when they compared the efficiency of using short and long read sequencing. So what they did is they compared if you spent the same amount of money on short read sequencing or on PacBio sequencing, and this is with the Sequel I system, then you did gene prediction, and you remove anything that has errors in it, and you dereplicate. You actually find twice as many genes by sequencing with PacBio than you do with short read technology. And that's because gene discovery is very dependent on contiguity. Okay, so because when you assemble short read data, so much of the data never makes it into the assembly. That's a lot of wasted sequencing, basically. Whereas with PacBio, almost every read has 5 to 8 genes on it. And so that makes for a very good return on your investment. You don't have to have enough coverage of low-valent species in order to find genes. And again, this was with the Sequel system. With the Sequel II system, the math is, of course, even better. So in some cases, though, it really is helpful to do a metagenome assembly. And some examples of that would be where, for example, you have species for which no reference exists because it cannot be cultured. And maybe the species is very impactful for human health, for example, and you want to understand differences in health outcomes that may be strain dependent. So you can take HiFi reads and you can assemble them, and they assemble very well to a very high contig N50. And then you can also bin these contigs, right, to assign them to the most likely pairs, right, in order to make complete genomes. And the bins also tend to score very highly. So in this case, this is just some of the high quality bins. You can see they have very high level of completeness and a very high consistency as well. So unfortunately, I can't tell you the names of the genomes because we don't have permission to share that information because it's a patient sample. But it's a human fecal sample, and there's exactly the bacteria in there that you would probably guess. So one question that we get is how much can you multiplex? And so we have a suggestion for how you can calculate that. So the first question is, what is the estimated abundance of the most rare species that you're interested in? So maybe, for example, we want to look at species that are 1% abundant or higher. And so if you have one smart cell, you can expect 1% of the reads from that smart cell to map to a species that's present at 1%. I think that makes pretty good intuitive sense. Assuming that you have a genome size that's average. So then the next question is, what is your goal with respect to that species? So if you just want to determine its presence or absence, then you don't need that much data. You can get by with maybe 100 reads or so. If you want to do gene profiling, then you should have 5% coverage, or sorry, 5-fold coverage of that species. And so you need about 2,000 reads. And then if you want to do a metagenome assembled genome, MAG, in that case, you want to have 20x coverage of that genome. And so there, I made a quick little reference table here. So this is based on the output from the sequel version 1 chemistry, so all these numbers will go up with the new chemistry. But you can see if you have one sample per SMART cell, you can do genome assembly for species that are as low as 1% abundance. And if you multiplex more, you can multiplex up to 3 and still do very good gene profiling for species that are that low. But you're probably not going to have enough data to do assembly in those cases. So it really depends on your goal. If you're looking to do like metabolic gene profiling, then all you really need to do is gene discovery, in which case this is plenty of data. But if you, in other cases where you really do want to develop a reference, because there's no reference that exists yet, then you need higher coverage and you probably don't want to multiplex in that case. So this is an example of a collaboration we did with a customer who did want to do genome assembly. And they wanted to sequence the gut microbiome of premature babies. And because premature babies tend to have, are at high risk of having sepsis, and what is known already is that when the preemie has B. breve in the gut, then this is connected to a better outcome. But has been no reference genome for B. breve, and so it was not understood whether different strains of B. breve were better or worse than other ones in terms of predicting the outcome. So they sequenced a number of infant guts, and from this data they were able to generate new reference genomes for 3 different strains. So they went from having no reference genomes at all to now having 3. And B. breve is very important with gut development because it's one of these bacteria that really is able to process the human milk oligosaccharides into the short-chain fatty acids, which then can actually be used by the rest of the microbiome and baby. So here's a second example. This is from a more complex community looking at assembly and gene finding, so kind of both of these applications together. So this is a study looking at the cow rumen. And the cow rumen is a topic of a lot of interest because everyone likes to eat beef. It's very tasty. But unfortunately, cows are very bad for climate change. They produce a lot of methane. And so there's a fair amount of research that's going into understanding the cow rumen and determining, is it possible to fix cows so that they're not destroying the atmosphere and we can still have delicious steaks. So in this experiment, Nick Watson, who I know normally does a lot of ONT sequencing, actually used PacBio to do this project. And he used, in addition to PacBio, Illumina, and one of the goals was to compare how these 2 different technologies can be complementary in studies of this kind. And then finally, after looking at sequencing technologies, he's added in Hi-C linkage data to see how that can add further levels of information about the community. So one of the more interesting conclusions from this paper, to my mind, is that Illumina sequencing is obviously less expensive, and it's very easy to collect large amounts of data. And so that's what they did. And with PacBio, they collected only about 1/3 as much data. But despite that, the Contig N100 here is a lot higher with PacBio, almost 3 times as high as with Illumina. And so even with less data, long reads just assemble better. And I think everybody is probably pretty aware of that from the work that has been done with PacBio on plant and animal and human genome assembly. Long reads are necessary in order to overcome the repeat structures in complex organisms. And really a metagenome is like a complex organism. So why does that matter? Why does it matter if you have a more contiguous assembly map? Well, it matters because gene discovery is highly dependent upon contiguity. And so having a better assembly allows you to do much better and much more robust gene finding. So this is an example of one of the gene families that they were looking for in the cow rumen. They wanted to understand if you feed the cows antibiotics, which all cows get antibiotics in the US, does the cow rumen develop antibiotic resistance? So if you use just short read sequencing, then you would have assumed that no, there's no antibiotic resistance in the cow rumen. It's fine. But when you sequence with PacBio, what you see is that actually there's a lot of antibiotic resistance in the cow rumen. It's just that the Illumina data is not sufficiently assembled that you can effectively do gene discovery on that data. And so it's really important to think about choosing the right tool for the type of science that you want to do. Finally, as the last part of the paper, they added Hi-C data to both of the assemblies, and then they went and tried to understand, can they connect viral and phage and plasmid DNA to the host species? And the answer is yes. Hi-C is a really powerful tool for determining what viruses live in what hosts. So this is one of the more interesting conclusions that they found is that some viruses are only found in one host, pretty exclusively, but that other viruses live in many different kinds of hosts, all different kinds of hosts. And so I would encourage you, if you're interested in this type of work, to look up the paper and read it. It's a lot more detail than can really be presented easily in 1 or 2 slides. Okay, so then I wanted to finally just do a little bit of information about our new microbial isolate assembly, because there are some updates there that are either coming or already out. So I wanted to briefly just make my pitch. I know a lot of people are doing draft assemblies for microbial genomes, and they feel that those are good enough. And they think that doing a long-read assembly is too expensive. But I would argue that it's actually very expensive to rely on draft genomes only. And the reason is because it's hard to draw the kind of conclusions that your research may depend on by relying only on draft genomes. So in this study, some researchers compared draft and closed genomes for 3 different Gingivalis strains. And they picked this because it was a prominent species in NCBI. And so there were a lot of examples to choose from. So they picked 3 strains, and they used the draft assemblies that were there, and then they sequenced PacBio. And then they compared their ability to annotate either the draft or the closed genome. And what they found, first of all, is that when you use a draft assembly, typically you spend a lot of time trying different assembly tools and then tweaking and adjusting the parameters on those tools in order to get the most continuous assembly possible. But they found that generally, if you choose the assembly that is the most contiguous, the most assembled, it is also the one with the most errors in that assembly. And that's bad because those misassemblies tend to truncate genes, and those truncated genes make it harder to do gene prediction. And they make it harder to identify the probable function of those genes. So in this case, when they had the draft genome, everything in the pink here, they were not able to determine what the function of that protein was. But when you use a closed genome, since these genes are more intact, they were able to identify domains that had specific functions that were known. And as a result of that, things moved out of the hypothetical pot and into one of these more specific functional groups. And so there was just a lot more information in the closed genome compared to the draft. And in addition, they pointed out that if you were looking to do specific types of study, then you really do need to have a closed genome. So one would be metabolic capacity. If you're trying to understand what genes or functions are present in a microbe, you need to be able to determine what the functions of the genes are. If you want to determine the phenotypic differences between closely related strains, again, if you can't identify the genes that are present in one gene and not in the other, then you're going to be at risk of doing sequencing and finding nothing. And then in addition, if you want to look at horizontal gene transfer, again, this depends on structural rearrangements between proteins. And if you're relying on a draft genome, you can't actually tell what order the pieces are in. And then finally, comparative genomics is where you might be sequencing, say, 100 microbes that are all from similar strains. If you have only one reference genome and the rest are draft, it's very difficult to compare one to the other in any sort of definitive way to understand how evolution is happening among those genomes. So the happy news is that long-read sequencing is less expensive than it ever has been, as you all know. With the Sequel II instrument, we will very soon have a new set of barcodes released, barcoded adapters, so that you can multiplex at up to 48 base pairs. And then in addition, we have a newly released microbial assembly pipeline, which I believe you all have SmartLink 8.0.
They're planning to upgrade. 8人对分析的，现在8人是，你们好像还没有，应该快，就在安排时间应该是。They're planning this on schedule, but not.
Not yet. Great. So then I will give you a preview of what is coming. So here's multiplexing of 48 microbes on the Sequel II instrument. You can see that all these genomes are mostly closing. In a few cases, there are several conjugates. Some of these genomes actually have multiple chromosomes in them, so you wouldn't ever expect to see just one. And the nice thing about doing this on the Sequel II is that you get very high coverage of all the microbes, even at 48 plex, which is really great if you want to do things like methylation analysis. And we have run methylation analysis in-house on 48 plex samples, and it works great on the Sequel II. So I'm going to cut the one slide. So I'm going to come back and give you a little bit more preview of the microbial assembly pipeline. So this pipeline is now been pulled out and is now separate from the HCAP4 or Falcon pipeline that you would use to assemble more complex eukaryotic genomes. And it has several features that are specific only for microbes. So for example, we analyze the data before it goes into assembly to look for chimeric reads, and then those are removed because we found that sometimes the presence of just one or two of these chimeric reads was preventing genomes from closing. We also have included a new plasmid assembly step. So just like before, microbial assembly uses seed reads, and it takes these seed reads to make a draft assembly. Now, we take all the raw data and we map it against the assembly, and anything that doesn't map, we extract that, and we put that into the plasmid assembly pipeline. So we attempt to find the plasmids within those remaining data. And when during the plasmid assembly stage, we use different parameters that work better for these much smaller extrachromosomal genomes. We also have changed the mapping used for this pipeline to a mapping tool that is circular aware. And the reason we've done that is because if the mapping tool believes that the genome is linear, then when you map, all of the reads map only to one side of the genome, and the other side ends up with low coverage, right? Because actually they're connected, but it doesn't know that. And when you have a circular aware mapping tool, then you have even mapping across the entire draft assembly, which means that you get very high accuracy across the whole genome. So this is a really nice feature, and I think another thing that distinguishes PacBio from ONT software. So with ONT, you rely upon Illumina data in order to do error correction. But Illumina data you're not actually able to map Illumina data to all parts of a microbial genome. If you were, you could get close genomes from just Illumina, but you can't. And so what that means is with ONT, you're not going to have even accuracy across the entire genome. Parts of the genome will have high accuracy because they were able to be corrected with Illumina, and parts of it will have low accuracy because no reads were able to uniquely map to those parts of the genome. And that means there's going to be compromised gene finding and gene annotation. So this even mapping and even correction is a unique feature for PacBio. Okay, so I hope I can convince you that in addition to being the gold standard for assembling microbes, that the Sequel II is also a really powerful and exciting tool to start to look at metagenomes in a new way, and at a very reasonable price point that I think will be exciting to a lot of your customers. And with that, I'm happy to answer any questions, or Beryl can also take questions.
可以用普通话问没问题。你问问线上的人说。
大家有什么问题，现在群里面的可以先问。不知道他们在。
他们是在讲话还是在打字上？
讲话，他们来个群。
真是都问不出来。
说话。
当然，他们那边有。
你那边都有是什么？等于他们那边是那种，就是像是我可以先问。
我可以先问。他刚刚推讲，大概应该是那中部说到90%要是个。那关于那个 ITS 或 CUS， 它是有什么直接的区别？
So he was asking about 16S and ITS. What's the rates we recommend? How many rates is enough? Like, what's our recommendation for that?
Yeah. So we don't have our own recommendation. We find that it really depends on how complex the sample is. So less complex samples, you don't need as many reads. For soil, you maybe need a lot of reads. So it depends entirely on how complex the sample is. And I would say that if you have a standard number of reads that you know is good with Illumina, that's a good place to start. On the other hand, since with PacBio, you have species resolution instead of just genus, what that can mean is that even though it looks like you may be maxing out with Illumina, with PacBio, you may need more coverage because things that look the same with V4 only are actually different with full length. Does that make sense?
Something like that. Human gut samples. At least you have like 10,000 CCS.
So, yeah, a lot of times, a lot of service providers in the US, at least, deliver 10,000 reads per human gut microbiome sample. So that's a good place to start. But we have not actually tried to sequence enough samples so that we can, estimate when you would start to see a plateau.
就像刚刚老的已经是比较难搞的，但是它中间这种比较复杂的，它大概像比较简单，当然是10000条路，然后10万条路。
1万条是
10000条路，旁边就像土壤，越复杂了。
So like for soil sample, do you have like a good start number of the rates to look at?
Yeah, so, if human microbiome has a few hundred species in it, soil